name's Baron Coilis, and I work at the European Variation Archive, which is essentially an open access resource for archival of genetic variants from any species. So I've just been working at the EVA for just over 3 years. Previously, I was a bioinformatician for strictly working on human data, which was interesting because I was strictly working with genome version 37. It was that stage where we were trying to essentially get all researchers to jump over to GCR 38. And with the genome, the version 37 is quite highly annotated. So now I've moved over to the European Variation Archive, and I'm working with all different types of species. I'm able to see sort of the different qualities of genomes and the different genomes of quite niche and specific species. They're quite interesting, to say the least. So this webinar will focus on what the EVA is and essentially the data related to our latest release, which came out sometime last month. I'll go through the slides, and again, there'll be time at the end where we'll go through the Q&A box and answer as many questions as we can. But do also, I'll also take note of some of the questions that have been left in the Q&A box, because they can also be passed on to our developers as well, and we can always get back to you in regards to answering some of those questions. But just to give you an idea of the flow of this webinar, we'll begin with an introduction to the EBI and the EVA. So this will cover our role in the EBI and how we interact with other resources at the European Bioinformatics Institute. I'll then move on to the EVA submission. I don't want to take too much time on the actual submission process itself. It's mainly just to see how data comes into the EVA and then what we actually do with that data itself. Further on, we'll then jump into the RSID clustering and remapping. So then how we actually assign these RSIDs to variants, and then how they can be referenced, whether it be in external databases or whether we're referencing variant data in publications. And then we'll move on to the release 3 data access. So the release 3 will be covered in the previous point, the RSID clustering and remapping. We will take a deeper dive into the actually accessing the release 3 data and what this data actually consists of, because it will be the first release. It is the first release that we have, where we have variants that have been remapped to a different genome assembly. And finally, we'll finish off with some final notes just to summarize everything that we've gone through. And it will also allow us time, as I previously mentioned, for any questions we can go through, or anything that you would like to be passed on to the EVA developer team. If you do take a look at the bottom during the slide, I've tried to leave the EVA helpdesk email. It will be me on the other end of this email. So if you do have any other questions outside of the webinar, please do drop an email and I'll pick them up as soon as I can. But just to kick things off with an actual introduction to the EVA and the EBI. So we act as a fair data resource, and we abide by the fair data principles, meaning that we ensure that the data that comes into the EVA is globally unique and persistent, and it can be queried using specific variant identifiers, those of which we'll be taking a look at further along in the slides. We also ensure that the data is accessible, meaning that it's universally accessible and it's open and it's free to access. We ensure that the data is interoperable, meaning that the reference data and the reference identifiers we're assigning can be used to query the variant data in other resources, and that the format we're actually working with and format we're accepting data in is also interoperable, meaning currently we're working with the VCF format. And we also ensure that the data is reusable, in that the data we're receiving, the abundance of metadata associated with this data is in good enough quality that anyone trying to reuse the data can replicate the same results. So where we reside, just some general information in this slide, in that the EBI is a biological data home for biological data services and research and training. and it's part of the larger consortium European Molecular Biology Laboratory. And we reside in the European Bioinformatics Institute as a permanent storage and accessioning service for genetic variation from any species. And this is our new slide, which details the different, sort of the organization of the teams within the European Bioinformatics Institute. So you can find us just in the genetic variation and disease section, along with some other confusing names from different resources, such as the European Genome Phenome Archive, and the European Nucleotide Archive is also in here as well. As you can see, for the genes, genomes, and RNA, reason being that the acronyms can get quite confusing, where the EVA, the European Nucleotide Archive is the ENA, and then we also have the European Genome Phenome Archive, which is the EGA. We actually interact with the different resources as well. So the ENA, we use the sort of the SRA data model of submission to provide project accessions. We also interact with Ensemble as they consume some variant data from our FTP to display onto the Ensemble website. We also interact with biosamples. So as you'll see is that the samples associated with projects are also broken to biosamples during this process. And we also interact with the Europe PMC to ensure that any of the data coming into the EVA also has the associated publication, so that when you do look at the study in the EVA, you can also see the associated publication with that study. And just a slide on some numbers we have. So currently, at the moment, we have around 3.4 billion variants across 227 species. We hold the genetic variant information, the metadata, so the study, experiment, analysis. The submitter actually details this to us when they submit the data to us, as in how did they process the data, what samples did they use. And then once the data is sent to us, we also add the annotation to this data, such as the functional consequence of the variant and the population frequency, which we calculate using the Ensemble's tool, the variant effect predictor. So this will just cover a quick overview of the actual submission process. I won't go into too much detail in the submission process, as this will just cover how we receive data and then what we actually do with the data in further slides. So we do start off with a submitter that's generated some data, and they have their data in VCF files. We also need the allele frequency data, or the genotypes. And then that's accompanied by a metadata file, which is essentially an Excel file that the user can have free text entry to enter information about their associated study. That's sent to the EVA helpdesk. It's made sometimes one of my colleagues on the other side, but most of the time I'll be on the other side to then run the data validation pipeline, which is also available, readily available on our GitHub. And it includes tools to ensure that the format of the VCF is valid and that the reference alleles provided for the variant can be mapped back to a relevant reference sequence. Once everything's passed in the data, we can say the data's fine. The data is then archived, and we provide a project ID, which starts off with this prefix of PRJEB, and usually a 5 to 6 digit number, and an analysis ID. So we can have a single project with multiple different types of analyses. And the project ID is mainly what you'll find will be provided back to essentially ensure that your manuscript is published. So you'll find that the journal editors will mainly ask that they need proof that the variant data and the raw data is archived in a permanent storage. So they'll ask for this project ID as a proof of submission. So then this step moves on to what we actually do after the archival process. So this was the original submitted VCF file. So we've now archived this VCF. We've provided back a project accession and analysis accession. This is an example of what the raw VCF file will look like. So we'll have the chromosome, the position, As you can see, the ID column is left blank in this example. And then we have the reference allele and the alternative allele, and we'll have some additional fields that are cut off in this example. But essentially, we just want to focus in this example that the ID field is currently missing any entries. So after the data is archived and we've provided these accessions, we then run the VCF file through our variance accessions database, which is a piece of code that then literally provides back the same VCF. It spits out the same VCF, but this time the ID field is now populated with these SSIDs. And these are the, we call these the accessions for the variance, the study accessions for the variance, the SSID, so they're unique to the study. So what I mean when I mention that the SSIDs are unique to the study is that any non-human variant submitted to the EVA will receive an SSID. But usually when you find you're going through a publication paper, you'll find that SSIDs are rarely mentioned or rarely referenced when we're discussing variant data. And you'll mainly find it's the RSID that's used within publications. And what an RSID is the collection of SSIDs collected and clustered into a single identifier. So for example, we can have a original study that takes a look at the Bos Taurus. It finds a variant in the Bos from mapped against the Bos Taurus genome. And it says, we've discovered a variant at chromosome 1, position 123, that's submitted to the EVA, and then we provide an accession of SS111. That study, that same variant can be reported in a different study from somebody else. And they could say, we've used the same reference genome and we found the same alternative allele, we found the same variant at the same position. That will be archived in the EVA also, and then we'll provide an accession of SS222. And you can see the same thing in the third example, that we'll receive this variant from this study, a different study, and it will provide the same reference sequence with the same chromosome location of the variant, and that will receive an SSID of 333. And then periodically, we archive these into, we cluster these into RSIDs, which are more readily referenced when we're referring to variant data. And then these three IDs would be clustered into this single RSID. So then how do we ensure that these references, these variant identifiers are preserved? And how do we work with them? And how do we reference them? And how do we ensure that we're all referring to the same variant data? This is going back to ensuring that what we're doing as a fair data resource is knowing that these identifiers we're providing can still be used and are still readily used within the community. And that if you take a look at the RSID provided by our database, you'll be able to find the same variant information in another database, such as Ensemble. So this is where we moved on to the variant remapping, which is a way to preserve these RSIDs through new genomes that will be coming out in the future. So for example, we have the original reference sequence here, and then we say that the original variant was provided on this reference sequence, and it was A allele, and the alternative allele was a C allele. And then we have the original variant, the flanking regions surrounding the variant. And then we used the Minimap2 software, which remaps the variant to the new assembly, but it also evidently changes the chromosomal location, because as we know on the new reference, it's not going to be exactly the same sequence. But it lets us know that the variant has been remapped from the old assembly to the shiny new assembly. But a good thing to mention is that the Minimap2 works by also observing the flanking sequence and ensuring that these are matching from the original reported variant. But as you'll see on the next slide, is that this does have, we sort of observed some variability between the different species. So we've also had a bit of a less mapping rate for certain species such as pig, which will take further investigation, and we're looking into an individual level. But we've done this for the majority of species that we hold in the EVA database. So this is just a snapshot of some of the success rates that we've had across different species, and how this method of mapping using the flanking region, how it essentially comes across throughout different species. So then thinking back to this example using the accessions we provide. So this original variant provided on this original reference sequence, for example, will be provided an SSID sort of SS1, but then the SSID is also remapped onto the new reference sequence. The new genome, but the SSID will remain the same. So then we come to a stage where we're essentially combining our clustering pipeline with our variant remapping pipeline. So we have SSID as previously mentioned, we have SSID SS1 remapped onto the new assembly, and it retains its original accession SS1. And then when we come to clustering these accessions, we now have these two, we have SS1 and SS2. So we have SS1, which was originally reported on this original genome, but has now been remapped to this new genome. And then, for instance, we have SS2, which has just been reported on this new genome, and then that will be clustered on the RS1. But we need to find a way to state that SS1 was originally reported on a different version of the genome. And we'll see how we do that via the FTP and RS release 3 access in further on within the webinar. So moving on to the actual data access and how we can show how this data has been remapped. So within the EVA, we have 4 methods of data access. We're not going to look at the first 2 methods because these look at variants on the EVA website, but don't actually look at the release data we're going to be taking a deeper dive into. So we do have a study in variant browser, which can be used to query variants studies individually, but we're actually going to look at the FTP and the REST API. So the FTP will allow us to have bulk data downloads where we can download all the RSIDs per species or per assembly or per study. But then we also have the R API, which allows computational methods of access to this data and allows you to essentially build queries into the scripts or code where you can download release data automatically from the EVA. So just moving on to the FTP. So the FTP can be accessed on our website. And it does have it begins with our different releases. So we've had 3 releases in the past with the relevant dates associated with them. So we have release 1, release 2, and release 3. So if we take a look at release 3, we can see that the data itself is then organized per species or per assembly. So we can take a deeper dive into the species. And for this example, I'll be looking at the cow species, or Bos taurus. And then when we look into this directory, we can see that the assemblies are actually organized by either the assembly name or the INST accession, which is provided by NCBI. So we have the 3 different types of assembly that we have for the Bos taurus species, but we also have the accession related to them. So for example, we can take the GCA for 003055.5, query this in NCBI, and it will come up with a relevant assembly. And we can see that it just relates, it's associated with Bos taurus UMD 3.1.1. And we can see the assembly accession here. And on the right side, we can go a bit further and we can see that NCBI also provide these identifiers for individual chromosomes. So we can take a look at the sequence report. Try to zoom in a bit more. And for example, we can see chromosome 3 is usually referenced as CHL3, but then we can see that our assigned molecule that NCBI has provided an section for is GK 00003.2. And it's just another way that we ensure that when we use these identifiers, that the same we're referring to the same sequence when we take a look at the data in different databases or across databases. So we've included both of these. We've included the assembly name itself, but also we've included the accessions you can use to query these assemblies. And what we're finding is we're attempting to find out which is more readily used. I mean, we do find that the assembly name, researchers prefer to use the actual assembly name, but then the identifiers associated with the assembly name, we always try to make sure these two are linked together so that if we do find someone that is using these accessions, then we know what assembly we're referring to. So if you want to click on one of the assemblies, so I've selected the one we previously looked at in NCBI, so the UMD 3.1.1, and we can see we have a number of different files available. So we don't just have a file containing RSIDs in use at the moment, but we have this file, which we'll take a look at the moment. So this is the assembly accession, and then the current IDs, which refers to the RSIDs that are currently in use and are currently used to reference variant data for this particular assembly. But then we also have the deprecated IDs. And what this contains is that this contains RSIDs that were previously assigned, but we no longer can find associated information, and we can't find enough evidence to suggest that this is indeed an RSID, this RSID is indeed a variant. But we still wanted to include this information because this RSID has been assigned in the past, and we want to ensure that this information is preserved for future research. The next file we have, I'll take a look at the previous file, this file in a moment, but just moving on to the merged IDs. So this also contains RSIDs, but this contains RSIDs that may have been merged into newer RSIDs. For any particular example, we do have RSIDs that are merged into different, we do have previous RSIDs that are then merged into further RSIDs that have been created as we progress with sequencing technology. And it's just a way to ensure that this information is still preserved and made relatively available. And then we actually have a mixture of the two, which is the merged and deprecated RSIDs, where an RSID may be moved into another RSID, and then the RSID that it was moved into is also no longer in use. And it could be for a variety of reasons. The main reason I mentioned was that we didn't find enough evidence that the previous RSID assigned was pointing that this was indeed a variant, because RSIDs have been being assigned for a number of years now. So we also needed to ensure that we made the RSIDs that we have gathering from all these years. We also wanted to ensure that we made that readily available, but we wanted to ensure that the data we are making available is still abiding by these FAIR principles. If it wasn't, we just wanted to ensure that it was still available via the FTP bulk access. But taking a look at just the RSIDs that are currently in use at the moment, I've done a quick download and we can take a look at what the actual file looks like. Oops. So we can see that this is the actual download of the file I've just downloaded into my terminal, so we can take a quick look at. So taking a look at one of the variant lines, we can see that we have the chromosome, the position, the current RSID that's been used. So this will be the RSID that's been referenced. And then we have the PRJ number. So this would be the project number that originally reported the variant. We also have the SSID. So this relates to the SSID clustered underneath the RSID. In this case, we only have one SSID clustered underneath this RSID. And we also have the VC field, the key value pair, which just lets us know what the sequence ontology is for this particular variant. I believe the code 001483 just lets us know that it's a single nucleotide variant, which we can see, which has gone from C to T. But this is generally how the VCF files for the release are provided back. So you can do a bulk access download and download all the RSIDs related to this particular species or assembly, and it will just come in a VCF file that looks similar to this. But that's a bulk access. So now we want to ensure that if we want to take a look at a particular variant of interest, how do we do that next? So we know that the FTP access, it allows download accessions per datasets per species, but we also provide the deprecated and merged RSIDs available. Before we've done this, actually, we sent out a survey just asking the community if we should actually include these deprecated and merged RSIDs. And the general consensus was that they should still be included. Because as I mentioned, even though they aren't readily used, it is sort of a history of the variant. And this information has come from somewhere, even though we can't find enough evidence to say that this is indeed a variant that has been, that this is a variant. But in the past, it has been assigned this RSID. So we wanted to ensure that the data was made available. And we also do the release, the releases are periodically. So aim to have a new release every 6 months. And the data is preserved throughout the releases. So this means that our latest release, release 3, does still contain the data from the second release and the first release as well. We also have the original submitted and SS accession files available per study. So if you do go onto the study browser, we won't take a look at that now, unfortunately, as we have to take a look at the just for the release data. But we do also provide SSIDs back per study, and that's also provided back via our FTP. Let's come back to the beginning. I do apologize. So I'm just going to move on to our So just moving on to our API access. So we do also have an API, which then allows computational methods of access to the EVA release data. So an example of this can be found on the EVA website. You can type in just by typing in EBI EVA. Click on the first link. Sorry, my keyboard's moving. Keeps on doing multiple keys, but mine will carry on. So on the homepage of the actual website, you can see the different things we have at the top. So we took a look at the the FTP access. So if you go onto the homepage, you can actually find the FTP access on the homepage in the search snip box. So you can search snips by individually by just querying this box here, or you can take a look at what we previously looked at just by accessing the FTP access here. As I previously mentioned, we do have the study and variant browser available. But if you go into the API page, you'll actually find some examples of how the API can be used, which we'll be taking a look at. So we can use the API, we can use these endpoints here to fetch all the variants in a segment region, or for example, to fetch all the information associated with an SSID, or all the information associated with the variant by its RSID. So the actual API page on the EVA website has examples of this. So taking a look using this endpoint, I'll try to zoom in a bit, sorry. So we can see that using this JSON output, So I've used this endpoint here. So the web services identifies the clustered variant. So I'm trying to take a look at this clustered variant here, which is 4455555728. So this would be the accession, and this block will provide the variant information for this RSID in regards to how its details on this assembly, which is actually the Bos Taurus genome we looked at, the UMD 3.1. But this is actually one of the previous versions. So we looked at 3055.5. This is 3055.3. And it will say it lands on this contig. So as we saw, the contig have identifiers, so that will relate to a specific chromosome, you can see. We'll find the start position, the type of variant it is, if it's been validated through our validation pipeline, and the actual date the variant itself was created. But then if you scroll further down to the second element, you can find that the same accession will provide additional details, but this time for a different assembly. So this will be the different Bos Taurus assembly. So as you'll see, the contig alias used has now changed because it's a different assembly. The start position has now changed. Before it was 877922. Now it's 675986 because it's a different assembly. The type of variant is still a deletion. It's been validated and the date it was created. And if you go even further down, you can see an entry for a third assembly as well. So the same RSID, so the RSID has been mapped to the to two additional assemblies. And we can see that we have a new assembly now for Bos Taurus that's been mapped to contig. Again, the contig ID has changed, the position has changed, but the additional details such as the type have not changed. But in this example, we can actually see this hasn't been validated, and this was because it was done. This was actually done during the relatively recently when we done the variant remapping. So this was only done this sometime last year. So this will also be checked to see if the validation service is indeed valid for this particular variant. So now that's how you can take a look at when you do query the API. If you are accessing the variant via computational methods, you can use these endpoints to bring up this data, and it will have multiple elements to just say that this is the original, this is the variant, and then this is the variant details as it's being remapped. But we can also add the history tag, which may not load, but we'll see how it goes. But I've saved it there. Yeah, it's got an internal error. Oh, come on. Either way, that will take a while to load. So I thought that may be the case usually when we get to working with the API query. So I've actually included the screenshot. I'm not sure if you can see, but it provides the same information, but give that a refresh again. It provides the same information as we just saw when we query a variant. But it also has additional information such as, there we go. So as you can see, it's provided the same information. So we'll go a bit further down. So we now have this section called operations. And what that lets us know is that it provides details of the history of RSID. So as we went through the FTP, we saw that we have RSIDs that have been deprecated, RSIDs that have been merged. This now lets us know that this RSID, the accession, and the reason for it to be moved to a particular, the reason it has been moved under a different RSID, or the reason it has been remapped, or what the case may be. It sort of gives us the history of the RSID and allows us to essentially preserve the information again that we're assigning to these variants. So for this reason, we can see that after remapping, the RSID mapped to the same locus. So in this case, the RSID was merged. If we go further down, we can see another example where we have this RSID split. And it says, due to a hash mismatch, the RSID that we originally had was issued to split from this another RSID. So what this lets us know is that if you are looking at a particular RSID and you're unable to find the information, do take a look at the history of the operations, because it may be the case that the RSID is no longer in use, or for any other reason, it may be that the RSID is also merged, or the RSID has been split into a different RSID. It's essentially attempting to provide back the variant information, but as we know, the variants can be quite complex, and there can be reasons why different things are affected for different reasons. So we need to ensure that we provide this, we preserve this information as we go along. Oops. So another thing I wanted to show is that the API is used for computational methods of access to the EVA release data. But we also readily have some Swagger documentation available, which provides a nice UI and a way to query the API and also give you the endpoints which you can use. So going back to the homepage of the EVA, we have the individual SNP search box. And then we also have the FTP access, which we saw. But then we also have the API access. So if you click on the API in this box, this will take you out to our Swagger documentation. And we can see that if we want to query these clustered variants now, that we can click on this section here. And then we could say, well, I want to query the, variant by its identifier. So we can click on the value here, so the clustered variant identifier. And then what this does is that it just allows us to provide the variant ID, and it will bring up the associated information that we have for this variant ID. So we can say try it out. We don't need to put in RS, so we can say we want to take a look at the information associated with RS123. So we can put in 123. Oh, bear with me. I'm having to use the keyboard. It's not being friendly from time to time. So we can take a look at RS123, hit execute, and that gives us pieces of information that we can see associated with it. So you can see that the information is provided back, similar to how we queried it using the browser. So we can see that we have this accession. The version is always the same. We always have the version 1 at the moment. The associated assembly in regards to its INSTC accession, the taxonomy, in this case, human, the config alias, the start date, the type, and if it's been validated, in this case, it hasn't. And we can also see the endpoint requests. So if we put this into our web browser, we will receive the same information that we got in the response body. But then we also provide the curl. It also provides the curl command, which you can use if you are trying to access it via a terminal or via a script. And this will provide the same data back, which is provided in a JSON format, and will provide the same information that is being provided in the response body. So I mean, the API it does allow programmatic access to the EVA, and it can still be used to it can also be used to query and do a bulk download. I mean, one of the main type of queries we find is that someone will want to query all the RSIDs related to this particular assembly, and they'll first do a bulk download of the the file provided in the FTP to grab all the RSIDs for that particular assembly, and then they'll write a script to do a quick sort of iteration through that file and update the curl command to go through these RSIDs and then grab all the information associated with these variants. So that is if individual researchers are trying to look at RSIDs in bulk, but then it also allows, if you are looking at variants at individual level, you can see that the operations does provide a history of the RSID in how it was originally assigned, and a bit more of why it may you find that if you are looking at this RSID, you can't find the associated information. The reason being for this is that the EVA actually took over the responsibility of assigning RSIDs towards the end of 2017. The original responsibility was actually done by dbSNP, but then to preserve the information, we had to import all the non-human RSIDs from dbSNP into the EVA. And we wanted to do this, but ensure that the RSIDs coming into the EVA abide by these fair data principles, in that if you are querying these RSIDs, you find the associated information with it. That file that we took a look at in the FTP, where we saw that we provided deprecated RSIDs. An example of these RSIDs is that some of the data we may have imported was provided as RSID years ago. And again, we can't find, there'll be instances where the information is missing. We don't know what reference assembly it was mapped to. But sometime in the past, this was actually provided an RSID. It was actually assigned an RSID. So we still needed to make this available. And potentially it could be something we could look at in the future and trying to sort of grab this historic data, grab this legacy data, and ensure that we can find how it was originally assigned an RSID and how it's still used. It's an attempt in trying to ensure that the history of RSID is preserved, while trying to ensure that the tools we're creating, such as the remapping pipeline and the clustering pipeline, work with future genome releases, because as we can see that more genomes are coming out, and we need to ensure that it works across species, and that when they have more genomes coming out, we can remap the variants from one genome to another with a relatively high success rate. You saw that in the graph that we didn't have as much of an success with the pig species as we saw between different assemblies. So this is something we want to address in going forward. We also need to ensure that we're talking to these different species communities and ensuring that the data we're providing is in an adequate format, is in a format that these species communities can use, and that the tools and the clustering we're doing is going in, we'll be able to, well, essentially what Pusi mentioned, we'll be able to work going forward as well. But just some final notes before we have some time for some questions. So the EVA itself, our base is that we act as a fair data resource for genomic variation from all species. We grant the accessions for the project analysis and the variants. This includes trying to preserve the variants, the accessions for project analysis and variants. The archive data is then clustered into these RSIDs, and the RSIDs are remapped to newer genome assemblies or different genome assemblies. And then that data that we do the clustering and remapping for is then made available through our FTP and API access. And with that, this is just a short slide just to say who our funding's from and the EVA team. And I'll want to finish up by saying thank you for registering and listening. Again, if you do have any individual questions, I know a bit of a short time to go through the clustering and remapping, but do leave questions, especially if you have particular use cases you would like us to look into. What we find a lot of the time is that we find users come to us saying, we have this RSID that was done in the past, and we can't find it within the database. And in this case, it's always good to look into our database, dig into the history of this RSID, and find what happened, what we're going to do with this RSID going forward, and what we're going to do with newer genomes that are coming out in the future. So thank you for listening.
Thank you very much, Baron. Thank you for today's webinar. And thanks everyone for joining in and asking your questions as well. So before we go on to Q&A, I just have a few announcements. So you can see our upcoming webinars at this link here in the slide. So just to keep you updated, so we have a webinar series which is starting next week from Tuesday for 3 Tuesdays in a row. It's about AlphaFold and its impact on research and development. And then, so today we heard about accessing genome sequences. So next week we have a webinar from PRIDE database, which is about proteomics data submission and access. So if you're interested, please register and join for that one. And then we also have a webinar coming up about COVID-19 data portal in 2 weeks' time. So please look out for that on our website. And we also record all of our webinars, just like we're doing it for today. And all the previous webinars are available at this link here in the slide. And just to mention that tomorrow, along with the recording, I will also share with you a link for the slides so that you can have a look at the slides while you listen to the webinar again. So all right, so with that, so thanks a lot, Baron. So we have a few questions already in Q&A box. So the first question is about the pipeline. Does the validation pipeline also look at quality of the data?
So no, the validation pipeline doesn't actually look at the quality of the data. It's something we want to establish that we're just an archive, so we need to ensure that the format is correct. But what we found is that sometimes we do receive submissions and there'll be a VCF file that's almost a terabyte in size. But majority of the variant lines, they're not actually reporting a variant. You'll find that all the samples provided in the variant lines are homozygous reference. So we sometimes do receive these sorts of files, but we've decided that it's not actually down to us to decide on the actual quality of the data itself. It was also in the VCF files, it may be that the case that the original submitter hasn't done any filtering, and that there may be some lower quality variant calls within the variant within the VCF file itself. But our role is just to make sure that we make the raw data available on the EVA website.
All right. So next question is about OWINE RSIDs. Can you check browser for OWINE RSID? It doesn't work for Tiziana.
Yeah, I can take a look at it. They're referring to the sheep.
Yes. Do you have any other update or something coming up what people should look out for from EVA?
No, it will mainly be again with the remapping pipeline. We're using the Minimap2 algorithm, which is it's working. As we saw, it did quite a bit of a success. But we sort of used that algorithm and didn't make any sort of specific changes to it at the moment, which is why we had a variable success with it. But yeah, it's for the next release, we're hoping to have a bit of more of an improvement in the remapping department.
Okay, so we have another question. How do you access metadata such as population frequency?
So this is actually done, it's something I didn't cover in the actual webinar, but it's actually done through the variant browser. So if you just go onto the EVA homepage, click on the variant browser, and the variant browser has some options on the left-hand side where you can filter your query, and then it displays the results on the right-hand side. But if you click on one of the variants where you query it, and then you go down, you can actually access it via these tabs here. So we'll have the files that the data is originated from, the genotypes for the study and the variant, and then the population statistics here. Well, in this case, they've just put the whole population and the allele frequency. But that's how you actually access the associated metadata for individual variants. For the actual study itself, you'll have to go onto the study browser, but we don't provide the population frequencies here. We just provide the general metadata. So then you would have to grab the project accession and then query that in the variant browser, which will provide that information. Thank you, everyone.